Good afternoon, everyone. Thanks for the introduction, and thank you to the organizers for inviting me here. So today I'm just going to give you a bit of information about the type of research that we're doing in my group at the moment on the genetic architecture of convergent coloration in lepidopteran mimicry complexes. So lepidopterans are butterflies and moths, so I'm mainly going to be talking about butterflies, but there'll be a bit of mothness going on as well. So as I was introduced, I'm Kanchon Dasmahapatra, and I'm from the University of York. So that's Old York and not New York, the original version. Anyhow, so I've got my name in big over there, but actually I'll start off by acknowledging the main contributors to this research. Yassine, who's a postdoc with me, and Ed, who's a PhD student with me, and Eva, who's doing a PhD at the Sanger. And this is basically the condensation of the body of work that they've been carrying out over the past 2 or 3 years. So convergent evolution. So what is convergent evolution? Convergent evolution is basically the repeated evolution of the same trait or the same phenotype in different lineages. And there are obviously lots of good examples of convergent evolution, and we had some examples over here in terms of the evolution of lactase persistence. But here are some other examples from the non-human world. Actually, there's a human example over here. So we've got different traits that have evolved multiple times. So for example, over here, we've got these shapes that have evolved in multiple lineages in, like, for example, sharks, dolphins, and ichthyosaurs. And they all look kind of similar. And that's because they've evolved in response to a common selective pressure, which is their aquatic environment in which these organisms are swimming. And this kind of shape gives them a kind of streamline shape, which allows them to swim through the water more efficiently. And we've got various examples over here, like echolocation, which has evolved multiple times. And it's interesting to think about the time scale over which these convergent phenotypes have evolved. In the case of the humans, obviously that's been a relatively recent event, but some of these other convergent incidences of convergent evolution happened over deeper evolutionary timescales. So in terms of the genetic architecture of convergent evolution, so what genes are controlling the convergent traits, so you can think about 3 major groups. One can have completely different genes controlling the same phenotype, so that would be falling into this category over here of divergent loci. We could have parallel evolution, so that's where the same gene is being used in multiple lineages and different species, but actually it's different alleles, so the different variants of that same gene has evolved independently in multiple lineages. Or we can have what's called collateral evolution. So here it's not only the same gene, but it's actually the same alleles at the genes that are being used repeatedly in different lineages. And that can happen due to either because the species are very closely related and they're still reproducing with one another, and the species can exchange alleles, so the alleles are being reused multiple times. Or it could be that this is kind of an ancestral polymorphism which is persisting in the species, and the same alleles are being reused. And you can kind of think about the time scale under which these 3 different modes of genetic architecture could operate. So this mode, where the same alleles, the same genetic variants are being used for the same phenotype, will presumably be among species which are quite closely related, because they still have to be reproductively compatible to be able to exchange genes. Whereas this type of mechanism perhaps might be more common where lineages have diverged a much longer time ago. So these are great examples of convergent evolution, but I would say that even more exciting and impressive examples of convergent evolution come from the world of Lepidoptera, so butterflies and moths. So this is a group of butterflies I work on. These are heliconius butterflies. They're found in the Amazon basin. So this over here, they all look pretty similar, but actually represent, I can't remember how many there are over here, there are 13 different species over here, and they have all evolved very, very similar color patterns. And that's because all of these butterflies are unpalatable to predators. So the main predators of these butterflies, or most butterflies, will probably be birds. And these color patterns, which are quite striking when you see them in nature, they're acting as warning signals to the would-be predators. So they're saying that you've tasted, a bird will basically try to sample these butterflies, and they'll get an unpleasant experience. They don't die from it, but they'll kind of feel unwell, and the birds will remember this color pattern and avoid that pattern. So essentially, all of these species are acting in concert to provide a consistent signal that the predators can remember. So this is a really great example of convergent evolution. I actually lied that these are heliconius butterflies. Those of you who are very keen-eyed might notice that this is actually a moth species. So moths and butterflies split about over 110 million years ago, and you can see that they've essentially converged on the same color pattern because of the protective advantage that's been offered by sharing the same trait. I'm not just going to show pictures of pretty butterflies. Actually, I will be showing quite a lot of pictures of pretty butterflies. So this is another example. So this actually is one heliconius species, Heliconius pneumata. This is an unusual species in that in one area you'll get multiple forms of the butterfly, and they've got a special genetic architecture for maintaining this polymorphism, and I'll talk about that in a bit. And these Heliconius pneumata forms are copying the appearance or mimicking the appearance of other species in a very distantly related group. So this is actually a different tribe of butterflies, and they're all different genus. And you can see that the amount of similarity between these quite distantly split species is quite remarkable. And actually, these are not the only members of this convergent mimicry ring. There's a huge range of different species which are essentially keying into the same protective phenotype. And this really is a fantastic system to understand the genetic basis of convergence. So, okay, one more great picture. So this is actually one species, Heliconius melpomene, and it's found all the way across Central America and into South America. And they're all the same species. They're completely interfertile, but they show different color patterns depending on where in South America you are. And that's because the local selective regime is slightly different. So for example, over here, the birds will recognize this color pattern, whereas out here, the birds are recognizing a different color pattern over here. And you get this kind of transition zone between color pattern forms. And that's something that I'll be using later on to understand the genetic basis of these traits. This here is another species in red, Heliconius erato. Again, overlapping distribution with this inner species, but wherever the two species are co-occurring or found in the same place, there you found this really amazing mimicry going on, a really excellent example of convergent evolution. So what's next? Right, so this is where all the action is taking place in our research group. So we've got South America over here. This is just to give you a bit of context as to what it's like. A lot of our work is done over here in Peru, Ecuador, and Colombia up here. This is a zoom in Peru, for example. This squiggly line over here is a small tributary to the Amazon River. It's already about 500 meters wide, so this is that river over here. This over here are the Andes, so they're going up to like 4,000, 5,000 meters. On this side is very dry. And this is the lowland Amazon basin. And this is where you find the greatest diversity, not just in kind of butterfly world, but in most other species. And these are the low foothills of the Andes, kind of 500 to 1,000 meters, where you get really spectacularly high biodiversity. So this is where we're doing most of our work. We catch butterflies like this with these nets. We really do literally run around in the jungle chasing them and catching them with the nets. They're actually relatively easy to catch because they're these chemically defended species, so they don't fly very fast because they're trying to show the color patterns to the predators. And this is just to show you what happens when you dress inappropriately in the Amazon and you get attacked by a wide range of different invertebrates. So these are the two main groups I'm going to be talking about. Actually, I'm going to mainly talking about this group over here. Heliconius butterflies, they're predominantly these types of color patterns, black and red and yellow. You get some of them which are these orange tiger patterns. And this is the Ithomine butterflies, much more diverse. Some of them have these orange color patterns, and a lot of them have these transparent colorations. Both groups, these maps are showing the diversity. The greatest diversity, as I was saying, is not in the Amazon basin, but actually in these foothills over here. So you can see that the highest diversity is in the foothills of the Andes. Both these groups of species are chemically defended. Heliconius butterflies over here, they gain their chemical protection from their caterpillars eating these vines, the passiflora vines, and they contain various cyanogenic compounds. The caterpillars kind of incorporate those poisons in their body, and the adults also become unpalatable due to those toxins. These butterflies, the Ithomine butterflies, are getting their alkaloids, pyrrolizidine alkaloids from a different group of plants. But again, that makes them unpalatable as well. This is showing you why some of them are called glass-wing butterflies, because they really are see-through. And we're trying to work on the transparency of these butterflies, but that's a different project. Okay, so what do we know about the color pattern genetics in butterflies and Lepidoptera? So a lot of work has been done on understanding the genetic basis of coloration in Heliconius butterflies. And as it turns out, it's only a small number of genes which I've put on over there, so cortex or ivory, optics, winte, and aristellus. And basically, those 4 genes, and in particular, this ivory and cortex, they're combining to produce this huge diversity of colorations. So it's very clear that in Heliconius butterflies, which has been intensively studied, that you're getting this mechanism causing this convergence. You're getting the same genes being used multiple times, but it's actually different alleles being used across these types of species. What we've shown, however, is that between closely related species, which still can occasionally interbreed in the wild, we find evidence that the species are gaining their similarity in appearance because they're exchanging the same alleles. So over here, for example, in these species are still reproductively compatible to a certain extent. They don't tend to hybridize much, but they do occasionally. But you can see that this pattern is popping up multiple times in this phylogeny. So these species are probably split about 1 million years ago or 2 million years ago. And this pattern, again, is coming up multiple times across this group of species. And we've shown in a number of studies that these convergent phenotypes that we find getting is a result of these species occasionally exchanging these adaptive alleles, and thus giving conferring their similarity in appearance. So at least in Heliconius, we've got 2 mechanisms that are predominating. We've got between closely related species, the same alleles or the same genes are being used, and between more distant related species, the same genes, but different alleles are being used. So what? Yeah, so we got this in Heliconius. But what we're wanting to know is, what is the genetic basis of this convergence across a much wider range of evolutionary timescales? So what do we see when we go beyond Heliconius? And what kind of mechanisms are responsible for this convergence? This kind of really is a great system for looking at this, because, for example, over here, we've got multiple species which are sharing the same phenotype. This is a Ithomine butterfly, and this is a day-flying moth. So these are species which split a long time ago. They're showing this convergence in color patterning. But also, you've got the system where within the same species, you've got genetic variation as well. And that allows us to get at to understand the genetic basis of the color patterning. So let's see what we got. So this is a summary of the species we're going to look at. You don't have to remember the names of these species. The main thing to take away from this slide is that we're looking at lots of different lineages. So we're looking at 1, 2, 3, 4, 5, 6 different species. Within each species, you've got this type of variation happening. They've got variation in the yellow 4-wing band. So you've got, within the species, some individuals have a yellow 4-wing band. Other individuals don't have the yellow 4-wing band. And you've got this happening between species that are very closely related, all the way down to species that are very distantly related. So we've got the same phenotype having been evolved over multiple different evolutionary timescales, which is allowing us to look at, does the genetic basis of convergence, at least in this system, vary over evolutionary time? So how do we do this? We actually use a very simple method. We just do genome-wide association analysis. As I mentioned before, you've got, in a lot of these species, you get geographic variation in this color patterning. So for example, in this particular species, this dark form is found in Peru and goes up into southern Ecuador. And there's a replacement with the yellow-banded form over here, which goes from northern Ecuador into Colombia. And in the middle, you've got this kind of hybrid zone. So you've got the two forms that are meeting, and there's basically hybridization happening in the middle. So essentially, there's lots and lots of gene flow between these two forms. And by genome sequencing individuals from this mixing zone, we can very easily get an idea of the genetic control. There's very little population structure, and we've got chromosomal references for these things. And we can do a genome GWAS using very small sample sizes, like 50 samples. I know people doing work on humans are looking at tens of thousands or millions of individuals. But in this case, because the genetic basis is generally a simple genetic basis, we can actually do it with very small sample sizes. And I'll show you the results. So this is the genome-wide association in this particular species, comparing these individuals with these individuals. So we're looking at this phenotypic variation. This is based on about 45 samples, I think. This is a standard Manhattan plot. So each dot represents a SNP. Each of these represents a different chromosome. And you can see over here, there's a single strong high peak of association over here associated with this phenotypic change. If we zoom in over here, so this is a zoom in of the peak, you can see very clearly a really, really narrow region of association. It's only about 5 kilobases wide or something like that. And the reason these SNPs are colored is that there's a group of SNPs, 1 or 2 SNPs, that are completely associated with the trait. So genetic variation at these SNPs is explaining 100% of the phenotypic variation in this system. What genes are around in this place? So this red arrow is indicating the start of the ivory gene. The ivory gene kind of extends in this direction over here. So we've got these falling in what we think is the intron of the ivory gene. And more about that in a little bit. So that's the comparison that I just showed you. But we've got other comparisons as well. So this is a comparison with another species, a different genus. Again, we've got a single peak of association. Mechanitis mesanoides, a single peak of association. And come on, this is the day-flying moth. Again, a single peak of association. So it looks like we've got a single gene explaining this same phenotype across all of these species. What's the gene? Well, this is a zoom in of it. I'm going to talk about this a little bit later on. In all cases, this peak is hitting the same gene. So the red arrow is again showing this ivory gene. So across all of these comparisons, the gene that is controlling this convergent phenotypic variation is the same gene, which is pretty remarkable. In the day-flying moth, we get this broad peak of association, which I'm going to come back to in a little bit. So we've got a little bit of functional validation of this ivory gene. This is an in situ hybridization chain reaction showing you the spatial expression of ivories. This diagram is not particularly clear, but essentially in this form of this species, you get ivory expression across the whole of the developing wing disc, whereas in individuals that look like this, you can get a very clear band where there's no ivory expression, and that's corresponding very clearly to where the yellow band would be in the adults. So we can show that ivory is indeed the gene that is expressed. It's causing this phenotypic variation. And we've got a little bit of CRISPR evidence over here, where if we knock out the ivory promoter, you get these white patches appearing in the butterflies. What's interesting is that so this is just zooming in on we've got these 4 species, and this is a depiction of where the genome-wide association peak is. This is the ivory promoter over here. The red lines over here are showing you the annotation for the ivory gene. What we find is that the peak of association is always in that first intron of ivory over here. But there's actually very little sequence homology at all. So if you look at these narrow regions, 5, 10 kb, if you try to do a sequence alignment, you don't find any homology between them at all. So it's very clearly across these comparisons, we've got this parallel evolution. The same gene is being used, but the alleles have been evolved independently in each of these cases. So what is ivory? It's a long non-coding RNA, and it is giving rise to this microRNA over here. and that microRNA is going on to repress various melanization genes. And if you want to find out more about that, there's this excellent paper that we did not write, but really does go into detail about what that ivory gene is doing. So what have I got to say next? So these are the species in which we did the GWAS. We looked for the difference in the genomes for the yellow and the lack of yellow band and the yellow band. But in each of these genera, that yellow band and orange band has evolved multiple times. So this gives us an opportunity to look for, is there evidence that these SNPs that we identify via genome-wide association analysis, are those SNPs being used repeatedly, i.e., is there evidence for the same alleles being used multiple times across closely related species, as we see in Heliconius. And we've thrown a huge number of tests at this kind of analysis. We've looked at actual SNP identity. We've looked at kind of blocks of descended common ancestry. In this system, we find no evidence of adaptive integration of the alleles, which to me came as a surprise, because generally we find evidence of this. So it looks like Heliconius, we do find lots of stuff going on over here, but actually outside of Heliconius, we're finding very little evidence of the same alleles being used. Instead, we find that the same gene is being used, but actually each species is evolving independently different alleles to achieve that phenotype. So I'm going to come back to this. So what's going on over here? Good. 3 minutes left, 15 seconds, 10 seconds by now. So I'm going to look at this a little bit more. So if we zoom out a little bit over here, this is a zoom out of the same GWAS. And what we find is that you get this very clear block of association with the phenotype. And this is completely associated with this phenotypic variation. And this sharp block-like shape is strongly indicative of this thing happening. We've got a genomic inversion over here. So we've got a 1 megabase genomic inversion, and that genomic inversion is perfectly correlated with this phenotype. So this is what's happening over here. And this means that outside the inversion, you get lots of recombination, but actually inside the inversion, between the two inversion haplotypes, there's no recombination, which allows perhaps the formation of supergenes. Whoops. So this is, again, the location of the ivory gene. So this is a great mechanism for maintaining a polymorphism in a population. And we know that this particular species is found in populations where you find both forms. So we've got this kind of inversion genetic architecture which allows the maintenance of a polymorphism in a population. What's really interesting is that when we look at another species, we find, again, the same inversion centered around ivory. We've got the same convergent evolution of this inversion architecture. And actually, we find it 3 times. So we've got 3 different lineages, very distantly related, all have independently evolved the same solution to maintaining a polymorphism in a population. So all 3 of them have got this inversion architecture, which allows them to maintain this genetic allelic variance at this ivory locus. So in conclusion, so we've got a whole load of comparisons over here, but we find that essentially we're finding the repeated use of the same gene ivory across 110 million years of evolution. And we find that our genome-wide association analysis is showing that we get these narrow regions of association in the first intron of ivory, but there's actually very little sequence homology. So we're currently trying to investigate what's going on over here, but presumably it contains some parts, something to do with transcription factors that are binding that affect the expression of ivory. And we find independent or parallel evolution of this inversion architecture, again, at the same gene. So we're getting multiple times the same genomic region and the same genetic architecture is being used. And in this system, at least, we find no evidence of integration of the ivory alleles among species. So that's all I've got to say. So we've got various people to thank. We've got the governments of the various countries to thank for providing the permits. Like I said, Yassine and Ed and Ava did almost all the work over here, and the range of collaborators over here from South America and the UK and Europe for me to thank. I thank you for your attention and listening to me, and I also thank the butterflies for giving up their lives for my science.